Beneath our feet lies an expansive biological frontier. The terrestrial deep subsurface contains billions of tons of living carbon embedded within dark crustal fractures. For decades, geobiologists assumed this deep biome operated on a uniform engine, abiotic hydrogen gas fueling methanogenesis and sulfate reduction. The logic relied on the potency of hydrogen as an electron donor and its frequent detection within deep fracture fluids. However, modern metagenomic sequencing reveals a significant data anomaly. Marker genes for hydrogen-driven methanogenesis are frequently absent or represent less than 5% of the microbial community. This evidence contradicts the universal hydrogen model. The data indicates that local mineralogy, rather than a single gas, dictates the apex metabolism of the deep Earth. Testing this localized model requires fluids strictly isolated from surface interactions. To find them, researchers utilize deep, open pit and shaft mines that penetrate kilometers into the continental crust. This industrial infrastructure allows direct physical access to ancient fracture networks safely. These shafts intercept paleometeoric waters and ancient groundwaters that have remained completely isolated for over 2 billion years. Data from these isolated voids reveals a biological landscape shaped entirely by local host rock, creating a mosaic of highly variable metabolic niches. Accurate modeling of these niches requires accounting for surface contamination brought down by groundwater movement. Topography and climate can rapidly alter the carbon sources in supposed subsurface portals like thermal springs. A single precipitation event can flush surface organic carbon deep into the fracture. This influx forces resident microbial communities to pivot from endemic autotrophy to surface-driven heterotrophy. This scatter plot tracks the carbon-13 isotopic composition of biomass relative to dissolved inorganic and organic carbon over time at the Bison Pool hot spring. Note the data recorded immediately after a major rain event. The sharp depletion in carbon-13 values signals a sudden reliance on surface-derived organic matter. The system returns to the historic baseline 8 days later, resuming its standard deep Earth autotrophic signature. To bypass these unstable transition zones, geobiologists utilize deep boreholes drilled directly into low permeability metamorphosed bedrocks. Because surface permeability skews metabolic profiling, valid deep biosphere models must rely exclusively on systems where the host rock chemistry dominates the hydrology. In these isolated systems, the specific mineralogy of the rock dictates the available energetic buffet. Consider the first geological test case, serpentinizing ophiolites, which serve as isolated mantle rock aquifers. Water reacting with olivine and pyroxene abiotically generates high concentrations of hydrogen gas and methane. Despite high thermodynamic potential from hydrogen, a lack of dissolved inorganic carbon creates a strict biological bottleneck. This constraint selects for specialized chemolitho-heterotrophs, adapted to carbon limitation rather than standard methanogens. The second test case is the iron-rich metasediments of the Deep Surf Observatory. Metagenomic sequencing of these fracture fluids shows a complete absence of the genetic markers required for methanogenesis. Instead, researchers identified a high abundance of the Psych2 genetic cluster, which facilitates extensive iron oxidation. Microbes here form mineral-hosted biofilms, physically anchoring to the rock to oxidize pyrite using dissolved nitrate. These two cases prove life in the deep continental crust entirely re-engineers its dominant metabolic pathways to exploit whatever specific minerals the local rock provides. To resolve the energy debate, researchers evaluated the thermodynamics of 15 distinct redox reactions across 5 separate deep mine laboratories. This dual-pane data visualization shows the result of calculating energy availability purely by the moles of electrons transferred in a reaction. On a purely molar basis, methanogenesis and sulfate reduction appear favorable, suggesting they are primary energy sources. However, the molar metric fails to account for the physical scarcity or abundance of these compounds within the actual fracture fluids. A more accurate metric is volumetric energy density, calculating the actual joules of energy available per kilogram of water. When adjusted for physical fluid volumes, the thermodynamic hierarchy reorders. Methanogenesis and hydrogen-coupled sulfate reduction drop to the lowest energy-dense options across almost all sites. The physical abundance of other minerals completely overwhelms the energetic potential of abiotic hydrogen. The oxidation of ferrous iron, sulfur, and sulfide emerge as the true energetic drivers. These results also show the consistent thermodynamic dominance of nitrate reduction across different environments. When quantified by volumetric reality, the deep biosphere functions as an iron, sulfur, and nitrogen-driven engine, rendering the hydrogen baseline mathematically obsolete. If methanogenesis provides negligible volumetric energy, we must explain why methane and methanogens are still consistently detected in deep aquifers. The Beatrix mine provides an answer through the inverted biomass pyramid model. Methanogens and methane-oxidizing archaea represent only a tiny fraction of the total population. These organisms function as carbon processors and byproduct generators rather than primary energy drivers. The sulfur waste they generate funnels upward to support a massive codependent microbial base. This base consists of thiobacillus bacteria, which execute this system's true dominant metabolism, the nitrogen and nitrate cycles. This architecture is a centrophy, where hydrogen-dependent microbes provide a support system for much larger sulfur-oxidizing populations. Ultimately, the presence of hydrogen or methane identifies just a minor metabolic cog within a much larger rock-driven engine. Broad, generalized assumptions regarding deep subsurface biology have been definitively disproven. Mapping deep ecosystem functions requires pairing local thermodynamic modeling with comprehensive endpoint proteomics. The terrestrial deep subsurface functions as a diverse collection of metabolic niches, where the local lithology dictates the unique chemistry of life.